Hello, my name is Kivilcim Ozturk. I work in Carter Lab at UCSD, and today I am going to be talking about our research titled Predicting Functional Consequences of Mutations Using Molecular Interaction Network Features. Variant effect prediction remains a central challenge for precision medicine. Missense variants are particularly difficult to understand as they change only a single amino acid in a protein sequence, yet can have large and varied effects on protein activity, spanning no difference to complete loss of function. Numerous tools have been developed to identify missense variants with putative disease consequences. Typically, these tools rely on protein sequence and structure information to predict variant effects at the protein level. However, biological function arises through higher order interactions among proteins called PPI networks. And PPI networks are known to be scale-free, meaning that they contain only a small number of high-degree nodes, so they are robust to random failure. But attacks targeting these high-degree nodes can be very disruptive to function. There are different measures for identifying these central nodes, such as degree, betweenness, closeness, or eigenvector centralities. And using these measures, we and others have shown that disease genes, especially known cancer driver genes, are significantly central in PPI networks, meaning that any mutations targeting these genes have the potential to perturb the network greatly. Based on this information, we sought to study variants not only by how they affect a particular protein, but also by how they impact the underlying molecular interaction network. The motivation being that different mutations targeting even the same protein might affect its interactions differently, perturbing different network regions, and resulting in different phenotypes. And the level of impact caused by a mutation to the underlying molecular interaction network may determine the likelihood of generating a disease phenotype. To do this, we need to be able to map mutations to 3D protein structures and interactions so that we can model their potential to affect network edges. So we collect the structural data on proteins and their interactions using PDB structures and homology models. And we annotated each residue as core or surface based on their relative solvent accessible surface areas. And then we identified all interface residues, which here are defined as a subset of amino acid residues that mediate physical contact between proteins. This way, we construct a structural resolved PPI network called SRNet, comprising more than 6,000 proteins and 10,000 PPIs. The structural annotation in this network allowed us to map various somatic mutations and germline variants to network edges to describe their potential to impact biological function. Here, core mutations are assumed to affect all interactions of the protein since they are destabilizing, while interface mutations only disrupt the specific interactions that the residue mediates, and the remaining surface mutations are assumed to have no impact on the network. After analyzing these variant-specific perturbed PPI networks, we saw that disease mutations, both somatic and germline, significantly target interaction interfaces. As can be seen here, somatic surface missense mutations in tumors are enriched at interface regions of both oncogenes and tumor suppressors relative to other genes. And it's the same for pathogenic germline variants compared to neutral variants. As these analyses support that both protein and amino acid level data derived from networks is informative about disease association, we hypothesized that network information would be useful for machine learning-based variant classification. So we designed 16 features describing network-level effects of mutations, including 7 protein-level features that estimate the significance of the target protein in the network. Then, depending on if the mutation targets a core, surface, or interface residue, we developed 9 residue-level features, which quantifies the impact of the mutation on the network architecture based on how it perturbs the network. Looking at the significantly different distributions of these network-based features for various types of variants, like driver versus passenger or pathogenic versus neutral, we can say that they show potential to distinguish between different classes of variants. We also compared these 16 network-based features to 83 non-network-based amino acid features that are classically used for variant classification, such as substitution effects on amino acid biophysical properties, evolutionary conservation of variant sites, local sequence biases, and site-specific functional annotations. We show that network features are not strongly correlated with these other classic non-network-based amino acid descriptors, which suggest that network features add new and useful information to the task of variant interpretation. Using all these 99 features, we trained a random forest classifier on cancer missense mutations designated as like the driver and like the passenger mutations. We performed 5-fold cross-validation with gene holdout to prevent information leakage and consequent overfitting. We trained different classifiers using different feature combinations to assess their utility in variant classification and demonstrated that addition of network-based features to classic amino acid level features significantly boosts performance. Then we evaluated the feature importances of the random forest classifier as the mean decrease in impurity and saw that network features, which here are highlighted with red, generally have higher importance than the classic amino acid features, which again supports the utility of network information for variant classification tasks. Another aspect to note here is that residue-level features perform well overall when focusing on interface residues only, which is where they are the most informative. So we further explored them by examining cases where the classifier was successful in differentiating between driver and passenger mutations occurring in the same proteins. And for these correctly classified same gene mutations, we observed a significant difference in distributions of residue-level features for the driver and passenger classes, which further supports that residue-level network features provide information useful for within gene mutation classification. Finally, we picked our highest performing classifier, which combines all network-based and classic amino acid features described so far. Then we assessed its performance relative to both cancer-specific and population-based methods. We evaluated performance on 4 external pan-cancer datasets constructed using different approaches, an in vivo screen, an in vitro assay, and 2 literature-derived datasets. Performance was assessed only on mutations that could be classified by all methods. Our classifier, named here as Network and AA, performed well based on the area under the ROC and precision recall curve scores across 4 benchmark sets and their mean relative to these state-of-the-art variant classification methods. You can see the rankings here on the right, and our method had relatively high performances overall. Accuracy, F1 score, and MCC measures also provided similar results, which further supports that network information is informative for variant interpretation, and the addition of network-based features to classic amino acid level features significantly boosts performance. So in conclusion, we developed network-based features that describe the relative importance of a protein within the network and the potential of mutations to alter the architecture of the network. We show that network-derived features can capture novel information about variant effects that is not already present in the classical amino acid features, that combining both sets of features improves classifier performance, and residue-level network features are helpful for distinguishing between variants within proteins. However, it's important to note that our method has a limitation of the coverage of mutations due to the requirement for structure and interface information. So in conclusion, our study suggests that information about molecular interaction networks can be incorporated into machine learning-based variant interpretation frameworks. This opens future directions for the development of novel features capturing network information. Now I want to thank my lab and my advisor, Dr. Hannah Carter, for all her help on this project and her amazing mentorship, along with all the current and past members of our lab. And I want to thank you all for listening.